Hello and welcome to Nighttime Science. Tonight we'll explore the fascinating realm of mushrooms and fungi, those soft-capped organisms that emerge from forest floors and hidden places, living lives that are slower and stranger than we might imagine. As you settle in, let your mind wander through underground networks and ancient partnerships, through glowing caps in dark forests and microscopic spores drifting on gentle breezes. There is no rush here. Only the slow unfolding of knowledge, one calm fact at a time. When you see a mushroom pushing up through leaf litter or growing along a fallen log, you are witnessing only a small fraction of something much larger. The mushroom itself, the visible portion with its stem and cap, is merely the fruiting body of an organism that lives mostly out of sight, threading through soil and wood in a vast, delicate network. This is the world of fungi, a kingdom of life entirely separate from plants, animals, and bacteria, operating according to its own quiet rules. Fungi occupy their own kingdom in the tree of life, distinct and ancient. They are not plants, though for many years scientists grouped them together. Plants capture energy from sunlight through photosynthesis, creating their own food from light, water, and carbon dioxide. Fungi cannot do this. They lack chlorophyll, the green pigment that enables plants to harness solar energy. Instead, fungi must obtain their nutrition from other sources, absorbing nutrients from their surroundings by secreting enzymes that break down organic matter externally, then taking in the resulting simpler compounds. This mode of feeding, called absorptive heterotrophy, defines their existence and shapes their role in every ecosystem they inhabit. The kingdom fungi is remarkably diverse, encompassing far more than the mushrooms we recognize. It includes yeasts, single-celled organisms that cause bread to rise and ferment sugars into alcohol. It includes molds, those fuzzy growths that appear on forgotten food, spreading in networks of branching filaments. It includes massive underground networks that can span acres of forest floor, and tiny microscopic species that live in partnerships with plant roots or inside the bodies of insects. Scientists estimate there may be between 2 million and nearly 4 million fungal species on Earth, though only about 150,000 have been formally described and named. The majority remain unknown to us, living quietly in soils, waters, and hidden niches across the planet. The fungi we call mushrooms belong primarily to two major groups within this kingdom. The first and most familiar is Basidiomycota, which includes the classic mushroom forms with gills or pores on their undersides, the shelf fungi that grow like brackets from tree trunks, puffballs that release clouds of spores when disturbed, and the mysterious stink horns. The second major group is Ascomycota, which produces spores inside tiny sac-like structures and includes morels with their honeycomb caps, cup fungi, and the highly prized truffles that grow entirely underground. Together, these two groups represent the vast majority of fungi that produce visible fruiting bodies, though many other fungal lineages exist that never form anything we would recognize as a mushroom. The structure of a typical mushroom is elegantly simple in appearance, yet each part serves a specific function in the organism's reproductive strategy. The cap, or pileus, protects the spore-producing surface beneath it. On the underside of the cap, depending on the species, you might find gills, narrow blade-like structures radiating from the stem to the cap's edge, or pores, tiny holes that are the openings of vertical tubes. Both structures serve the same purpose, providing surface area for spore production and release. The stem, or stipe, elevates the cap above the substrate, positioning it where air currents can catch the spores and carry them away. Some mushrooms have additional features like rings around the stem, remnants of a protective veil that covered the young gills, or a cup-like structure at the base called a volva, the remains of a membrane that enclosed the entire young mushroom like an egg. But these visible structures, the mushrooms we see and recognize, are not the organism itself. They are temporary reproductive organs appearing when conditions are right, releasing their spores, then withering away. The actual organism persists underground or within whatever substrate it inhabits, as a network of thread-like filaments called hyphae. Collectively, this network is called the mycelium, and it represents the true body of the fungus, the part that grows and feeds and can persist for years, decades, or even millennia. Each individual hypha is microscopically thin, often just a few micrometers in diameter, but they grow and branch and interweave, creating networks that can be astonishingly vast. A single cubic inch of soil can contain miles of these hyphal threads, invisible to the naked eye, exploring every tiny crevice and particle. The mycelium is the fungus's interface with the world, the surface through which it secretes enzymes to break down complex organic molecules and through which it absorbs the resulting nutrients. It is a slow, patient, thorough way of life, quite different from the active hunting of animals or the rooted stillness of plants. Understanding that mushrooms represent just one brief phase in a fungal life cycle helps us appreciate how different these organisms are from the plants and animals we know more intimately. A mushroom's appearance is an event, a response to environmental conditions, a moment of reproduction before returning to the hidden work of growth and decomposition. In this way, fungi embody a kind of patience, a way of existing that is unhurried and persistent, perfectly suited to our quiet exploration tonight. As we continue our journey through the science of mushrooms, we'll discover networks that connect entire forests, spores that can survive the vacuum of space, mushrooms that glow with their own light, and partnerships between fungi and other organisms that have shaped the history of life on Earth. But we'll take our time, moving slowly through each discovery, letting the information settle gently, like spores drifting down through still air onto the forest floor. Beneath your feet, whenever you walk through a forest or across a meadow, stretches an invisible realm. The soil teems with life we cannot see, and among the most extensive networks down there are the mycelia of fungi, reaching through darkness, connecting, exploring, slowly expanding in their silent search for nutrients and opportunity. To understand mushrooms, we must first understand this hidden foundation, this gossamer architecture that represents the true body of the fungus. Mycelium consists of hyphae, those thread-like filaments we mentioned earlier, each one a tube of living cells extending at its tip, branching and rebranching as it grows. The hyphal tip is where growth happens, where the fungus explores new territory. Behind the advancing tip, the older portions of the hypha may die back or persist, maintaining the network's integrity. This growth pattern allows mycelium to be remarkably efficient, concentrating resources at the exploring edges while maintaining connections to areas already exploited for nutrients. These hyphae are not simple tubes. In many fungi, they contain multiple nuclei within each cell, and the walls between cells have pores that allow cytoplasm and even nuclei to flow between compartments. This creates a kind of shared internal highway system, where resources gathered in one part of the mycelium can be transported to fuel growth elsewhere. When a portion of mycelium encounters a rich food source, perhaps a fallen branch laden with available nutrients, it can signal to other parts of the network, which may then redirect growth toward that abundance. This is not conscious decision-making, but rather a sophisticated biochemical response system that allows fungi to act in surprisingly coordinated ways despite having no brain or central nervous system. The mycelium's thread-like architecture gives it an extraordinary surface area relative to its volume. Every section of every hypha is in intimate contact with its environment, whether that environment is soil, wood, leaf litter, or living plant tissue. This maximizes the fungus's ability to secrete digestive enzymes into its surroundings and absorb the nutrients released by those enzymes. It is a form adapted perfectly to the fungal way of life, patient and thorough, extracting nourishment from sources that might seem unpromising to other organisms. Some mycelia remain relatively small and localized, perhaps growing through a single fallen log or occupying a patch of soil just a few feet across. But others grow to sizes that challenge our intuition about what a single organism can be. The mycelium continues growing year after year, expanding outward in all directions from where it first germinated from a spore as long as conditions remain favorable and food sources persist. In the Malheur National Forest in Oregon, scientists have documented what may be the largest known organism on Earth by area, a single individual fungus of the species Armillaria ostoyi, commonly called the honey mushroom. This organism covers approximately 2,400 acres of forest floor, its mycelium spreading through the soil and attacking the roots of trees, acting as a pathogen in this case. Genetic testing confirmed that the mushrooms appearing across this vast area all belong to the same individual, all connected through one enormous underground network. Estimates suggest this individual fungus weighs somewhere in the range of 7,500 to 35,000 tons and may be between 2,000 and 8,800 years old. It has been growing since before written history, slowly expanding through the forest, outliving generations of trees, persisting through fires and droughts and the turning of countless seasons. Its growth is imperceptible on human timescales, adding perhaps a few feet to its radius each year. But that patient accumulation across millennia has created something immense. This humongous fungus, as it has been called, illustrates the fungal strategy of persistence and gradual expansion. Unlike animals that must actively hunt or forage for each meal, or plants that must compete for space in the canopy, the mycelium simply grows, explores, and absorbs. When it encounters a tree root, it can invade the root tissue, extracting nutrients and eventually killing the tree. The dead tree then becomes another food source, further fueling the fungus's expansion. It is a slow-motion drama playing out beneath the visible forest, shaping the ecosystem in ways that were invisible until scientists began investigating the genetic relationships between distant mushrooms. Not all mycelia are parasitic like the honey mushroom. Many form beneficial partnerships, which we will explore more deeply soon. But even those mycelia that simply decompose dead material, breaking down fallen logs and leaf litter, can grow to impressive sizes. A mycelium decomposing a large fallen tree might extend through the entire length and breadth of that log, following the wood's internal structure, breaking down cellulose and lignin molecule by molecule year by year, until the solid wood becomes soft, crumbly, and eventually returns to soil. The way mycelium grows and branches creates patterns that are almost fractal in nature, with larger branches giving rise to smaller branches which divide into still finer filaments. This branching pattern is not random but responds to the environment. Where nutrients are abundant, branching increases, creating a denser network. Where nutrients are scarce, growth may be more directed, with hyphae extending further before branching again, searching for better conditions. The mycelium, in its simple way, maps the nutritional landscape of its environment through its growth pattern. Observing mycelium directly requires careful excavation or special techniques. If you gently disturb the leaf litter in a forest or pull back bark from a long-fallen log, you might glimpse mycelium as white or colored threads, sometimes forming dense mats that look almost like cotton or cobwebs. In laboratory conditions where fungi are grown on agar and petri dishes, the mycelial growth becomes visible as the colony expands outward in a roughly circular pattern, the margin of the colony representing the actively growing hyphal tips. This hidden mycelial network is fundamental to how fungi interact with their world. It is the feeding structure, the exploration system, the connection between past and future. When environmental conditions become favorable, when temperature and moisture reach the right levels, the mycelium can divert resources to form fruiting bodies, those mushrooms we see above ground. But the mycelium itself continues, unchanged in its essential nature, whether producing mushrooms or not. It is the constant beneath the ephemeral, the enduring structure that can persist for lifetimes while mushrooms come and go in a matter of days. Some fungi produce specialized structures within their mycelium, such as rhizomorphs, which are thick cords of densely packed hyphae that can transport water and nutrients across longer distances more efficiently than individual hyphae. These structures look like dark shoelaces or roots and can grow through soil to connect distant patches of mycelium, or to help the fungus spread to new areas. The honey mushroom, that massive Oregon specimen, uses rhizomorphs to move through soil and attack trees some distance from its main body. The mycelial network also serves as a kind of living archive. As it grows through complex environments like forest soil, it encounters and interacts with countless other organisms, bacteria, other fungi, tiny animals, plant roots. These interactions can influence the mycelium's growth and chemistry, and in some cases the mycelium forms lasting associations that define new ecological relationships. The most important of these relationships, the mycorrhizal associations between fungi and plant roots, depend entirely on the mycelium's ability to extend far beyond where the plant's own roots can reach, exploring a much larger volume of soil and bringing back nutrients to share. As we contemplate this hidden world of mycelium, we can appreciate that forests and meadows have layers of complexity we rarely consider. Above ground, we see trees and flowers, insects and birds, all the vibrant, visible life. Below ground, extending through the darkness, mycelial networks connect and communicate, decompose and transport, forming partnerships and competing for resources in a realm where success is measured not in moments but in years and decades. It is a slower world down there, but no less dynamic, no less essential to the functioning of the ecosystems above. The mycelium reminds us that much of what matters happens out of sight, that patience and persistence can achieve what speed and force cannot, that being connected to food sources, to partners, to the earth itself is a viable strategy for survival. As your mind follows these branching threads through imagined soil, let yourself settle deeper, becoming quiet and still, content to exist in this moment without urgency, like the mycelium itself, simply being, simply growing, slowly and steadily through the dark. One of the most remarkable discoveries in forest ecology over recent decades has been the realization that trees are not isolated individuals but are connected through underground fungal networks that facilitate communication and resource sharing. Scientists have called this system the Wood Wide Web, a vast network of mycorrhizal fungi that link plants together in ways that fundamentally change how we understand forests and plant communities. Mycorrhiza means fungus root, and mycorrhizal associations are partnerships between fungi and plant roots that benefit both organisms. The relationship is ancient, dating back more than 400 million years, to when plants first colonized land. In fact, many scientists believe that plants could not have successfully made the transition from aquatic to terrestrial environments without fungal partners to help them extract nutrients from primitive soils. These partnerships have shaped the evolution of both plants and fungi ever since. There are several types of mycorrhizal associations, but the two most common and ecologically significant are arbuscular mycorrhizae and ectomycorrhizae. Arbuscular mycorrhizae are the more ancient form, found in approximately 70 to 90% of plant species, including most agricultural crops and herbaceous plants. In this relationship, the fungal hyphae penetrate the plant root cells and form branching structures called arbuscules that look like tiny trees inside the cell. These arbuscules are the sites where nutrients are exchanged between fungus and plant. The fungus receives sugars and other carbon compounds from the plant, products of photosynthesis that the plant creates using energy from sunlight. In return, the fungus provides the plant with mineral nutrients, particularly phosphorus and nitrogen, that its mycelium has gathered from the surrounding soil. Ectomycorrhizae are found primarily in trees, especially in temperate and boreal forests. Species like pines, firs, oaks, birches, and beeches form ectomycorrhizal partnerships. In this type of association, the fungal hyphae do not penetrate the plant cells, but instead form a dense sheath around the root tip and grow between root cells, creating an extensive interface for exchange. Many of the mushrooms we see in forests, including boletes, chanterelles, and russulas, are the fruiting bodies of ectomycorrhizal fungi. These mushrooms represent only the reproductive moment of fungi whose mycelia are intricately intertwined with tree roots throughout the forest. The benefit to the plant is substantial. A plant's roots can only explore a limited volume of soil, constrained by the energetic cost of growing and maintaining root tissue. But the fungal mycelium extends much further, with hyphae that are far thinner than even the finest root hairs, able to penetrate smaller soil pores and explore a vastly larger area. The mycelium of a single mycorrhizal fungus can connect with multiple plants simultaneously, and a single plant can host multiple species of mycorrhizal fungi. This creates a complex network where many plants and many fungi are all connected, all exchanging resources through countless underground connections. Research has shown that these networks do more than simply move nutrients from soil to plant. They also facilitate movement of resources between plants. In forest studies, scientists have traced carbon isotopes and found that sugars produced by one tree can travel through the mycorrhizal network and be taken up by another tree. This means that trees are sharing resources, that a large tree with extensive canopy access to sunlight might be supporting smaller trees growing in its shade, sending them carbon through the fungal network. Mature trees have been found to support their own seedlings this way, and even to support seedlings of other species. This resource sharing is not always balanced or altruistic in the human sense. The fungi are not consciously redistributing resources for the good of the forest. Rather, the network operates through supply and demand gradients. Fungi move nutrients toward areas of their mycelium where conditions favor growth, and carbon flows toward fungi from whichever plant is providing it most generously. but the result is a system where resources can be redistributed across the forest, where trees in good growing conditions can effectively subsidize trees in poorer conditions, mediated by the fungal network between them. Even more intriguing, research suggests these networks can carry chemical signals as well as nutrients. When a plant is attacked by insects or infected by disease, it produces defensive chemicals and may also release signal molecules. There is evidence that these signals can travel through mycorrhizal networks to neighboring plants, which then begin producing their own defensive compounds even before they are attacked. This would constitute a kind of early warning system, allowing plants to prepare their defenses based on what is happening to their neighbors. The concept of mother trees has emerged from this research. These are the largest, oldest trees in a forest, which have the most extensive root systems and mycorrhizal connections. Mother trees serve as hubs in the network, connected to many other trees and acting as sources of stability and resource distribution. When these large trees are removed through logging or clearing, the network is disrupted, potentially affecting the health and resilience of the entire forest community. The implications for forest management and conservation are significant and are still being explored and debated. The Wood Wide Web is not a centralized or designed system. There is no command center, no coordinator ensuring fair distribution. It emerges from the countless individual interactions between fungal hyphae and plant roots, each partnership operating according to simple biochemical rules, the exchange of carbon for nutrients. Yet from these simple interactions arises complexity, redundancy, and resilience. A forest connected by mycorrhizal networks may be better able to withstand drought, as water and nutrients can be shared. It may be more resistant to disease as defensive signals spread through the network. It becomes, in some sense, more than a collection of individual trees, instead forming an interconnected community. Not all fungi in this network are beneficial partners. Some are cheaters, species that connect to the network and take resources without providing benefits in return. Some fungi are parasitic, taking more from plants than they give. The system includes competition as well as cooperation, and the balance between these forces shapes forest ecology in ways scientists are only beginning to understand. The mycorrhizal network also connects plants to the decomposition system we mentioned earlier. Some fungi can act as both mycorrhizal partners and decomposers, breaking down dead organic matter to extract nutrients and then transferring some of those nutrients to living plant partners. This creates a direct link between the dead and the living, a way for nutrients locked in dead leaves and wood to be recycled back into living trees, mediated by fungal networks. Different plant species have preferences for different mycorrhizal partners, and these preferences can influence which plants can grow successfully in which locations. Some highly specialized orchids, for example, can only grow in the presence of specific fungal partners, and their seeds will not even germinate without the fungus present. This creates intricate webs of dependency, where the presence or absence of particular fungi determines which plants can survive, which in turn affects which animals can find food and habitat, rippling out through entire ecosystems. The Wood Wide Web reminds us that forests are not just collections of trees, but are integrated systems where communication and cooperation occur in forms quite different from what we see in the animal kingdom. There are no words or gestures here, no visual signals or sounds, but rather a biochemical conversation conducted through miles of mycelial threads in the darkness beneath the forest floor, slowly and continuously over seasons and years. As we reflect on these underground networks, we might consider how connection and interdependence are fundamental to living systems. how what appears isolated and individual is often part of larger patterns of relationship and exchange. The trees stand separate above ground, each with its own trunk and crown, but below in the soil they are joined, sharing resources and information through fungal intermediaries that make the forest function as something approaching a single living system. This is knowledge that settles quietly into understanding, requiring no immediate action, simply expanding our appreciation for the complexity hidden in familiar places. The next time you walk through a forest, you might pause and consider what is happening beneath your feet. The silent communication and exchange, the slow pulse of nutrients moving through living threads, connecting tree to tree in an ancient partnership older than flowers, older than birds, as old as forests themselves. The life of a fungus begins with a spore, a microscopic package of genetic material and minimal resources cast into the world with no guarantee of success. Spores are how fungi reproduce and disperse, how they colonize new territories and ensure their lineage continues. The journey from spore to mature mycelium to mushroom and back to spore again is a cycle that has repeated countless billions of times across the history of life on Earth. And tonight we will follow that journey in its quiet, patient unfolding. A single mushroom can produce an astonishing number of spores. The numbers vary by species and by the size of the fruiting body. But even a modest mushroom can release millions of spores, while larger specimens may release billions or even trillions over their brief reproductive lifetime. These spores are produced on the gills or within the pores of the mushroom, on specialized cells called basidia, in the case of Basidiomycota, or within sacs called asci in Ascomycota. Each spore is incredibly tiny, typically just a few micrometers across, barely visible even under a light microscope, small enough to be carried easily by the slightest air current. The production of such enormous numbers of spores represents a significant investment of the fungus's resources, but it is necessary because the odds of any individual spore successfully establishing a new colony are vanishingly small. Most spores will land in unsuitable locations, places too dry or too wet, too hot or too cold, or where the necessary nutrients are absent. Many will be consumed by other organisms, bacteria, or tiny animals that feed on such microscopic particles. Some will germinate, but then fail to find a compatible mycelium to mate with, because most fungi require genetic material from two different individuals to complete their life cycle. The strategy, then, is one of overwhelming numbers, producing so many spores that even if the success rate is 1 in a million, some will survive and establish new colonies. The mushroom employs various mechanisms to release its spores into the air, where they can be caught by breezes and carried away. In many gilled mushrooms, the spores are forcibly ejected from the basidia through a mechanism involving a small droplet of fluid that forms at the base of the spore. Surface tension creates forces that suddenly launch the spore into the narrow space between adjacent gills, where it can then fall freely and be carried away by air currents. This ejection happens on a microscopic scale but represents a precisely controlled biological mechanism refined over millions of years of evolution. Interestingly, mushrooms can create their own air currents to aid in spore dispersal. As moisture evaporates from the gill surfaces, it cools the air immediately around the mushroom, creating a gentle downward flow of cool air that then spreads outward along the ground. This convection current helps carry spores away from directly beneath the mushroom cap, increasing the chances they will be caught by larger air movements rather than simply falling straight down to the ground directly below. Even in perfectly still air, this self-generated airflow can carry spores several centimeters from the mushroom, enough to escape the immediate vicinity and have a chance of dispersal. Once airborne, spores become part of the atmosphere's particulate load. They drift with winds and weather systems, rising and falling with thermal currents, traveling distances that can range from a few meters to potentially thousands of kilometers. Fungal spores are found in air samples taken from high altitudes and from the middle of oceans, far from any land. They are part of the biological component of clouds and may even play a role in cloud formation and precipitation as particles around which water vapor can condense. Spores are remarkably resistant to environmental stresses. Their tough cell walls, often containing melanin and other protective compounds, shield the genetic material inside from ultraviolet radiation, desiccation, and temperature extremes. Some fungal spores have been shown to survive exposure to conditions that mimic outer space, intense ultraviolet radiation, vacuum, extreme cold, and cosmic rays. In experiments where spores were exposed to stratospheric conditions or even sent outside spacecraft, a small percentage survived and remained viable. This extraordinary resilience raises interesting questions about whether fungal spores could potentially survive transport between planets, though actual interplanetary transfer remains purely speculative. The ability to survive harsh conditions means spores can remain dormant for extended periods, waiting for suitable conditions before germinating. In the right environment, perhaps in soil or on wood where moisture and nutrients are adequate, a spore will absorb water, swell, and begin to germinate. A tiny germ tube emerges, the beginning of a new hypha, and starts to grow and branch, exploring its microscopic environment. This young hypha is vulnerable, requiring continued favorable conditions to survive. It must find food, must avoid being consumed by bacteria or microscopic grazers, and in most cases must encounter a hypha from a compatible mating type to form a complete reproductive mycelium. Fungal mating systems are diverse and complex, but many species require the fusion of hyphae from two genetically distinct individuals before they can complete their life cycle and eventually produce mushrooms. The young hypha growing from a spore represents only half the genetic equation. When it encounters a compatible hypha from another spore, the two can fuse, and their genetic material combines. The resulting mycelium contains nuclei from both parents and can grow into a mature colony capable of producing fruiting bodies. This requirement for genetic combination from two sources helps maintain genetic diversity within fungal populations and may reduce the risk of inbreeding. Not all dispersal is by air. Some fungi have evolved to use animals as dispersal agents. Truffle fungi, which produce their fruiting bodies underground, emit powerful aromas that attract animals like pigs, deer, rodents, and various other mammals. These animals dig up the truffles and eat them, and the spores pass through the digestive system undamaged, being deposited in feces at some distance from where the truffle was found. This provides both dispersal and a small package of nutrients to help the spores establish in their new location. Other fungi produce sticky spores or spore masses that adhere to animals, either deliberately through structures designed to catch onto fur or feathers, or incidentally as animals brush past spore-laden fruiting bodies. Insects, particularly flies and beetles, are important dispersal agents for many fungi, attracted by odors or by the mushroom structure itself, which may provide food or habitat. Some fungi have fruiting bodies that forcibly eject entire masses of spores. Puffballs, when mature, develop a hole in the top through which clouds of spores puff out when the fruiting body is disturbed by rain, wind, or passing animals. The Earth star fungi have similar mechanisms with structures that change shape in response to moisture changes, creating bellows-like actions that expel spores. These mechanisms harness environmental forces, turning raindrops and wind gusts into opportunities for dispersal. In aquatic and semi-aquatic environments, some fungi produce spores adapted for water dispersal, with shapes or structures that help them float or move with water currents. These fungi play important roles in stream and lake ecosystems, decomposing leaves and wood in water and providing food for aquatic invertebrates. The spore stage represents both an end and a beginning, the completion of one generation's reproductive effort and the potential start of the next. In this microscopic form, the fungus is at its most vulnerable but also its most mobile, able to cross distances that the immobile mycelium could never traverse. The spore is a package of patience, able to wait through adverse conditions and a vehicle of exploration, capable of founding colonies in new and distant locations. As you consider the vast numbers of spores released into the air every moment of every day, from countless mushrooms in forests and fields around the world, you might imagine the atmosphere as containing this invisible component, this living dust of potential, each particle carrying within it the possibility of becoming a new organism, a new network spreading through soil or wood, perhaps one day producing its own mushrooms and releasing its own spores to continue the cycle. The scale is vast, the numbers incomprehensible, yet each individual spore is a complete unit, a self-contained possibility, drifting on currents too gentle to feel, settling in places random and numberless, and occasionally, rarely, finding conditions just right for life to begin again. This is the quiet mathematics of survival, played out in microscopic scale across geological time, the patient expenditure of resources on countless chances in the hope that a few will succeed. There is something calming in that acceptance of uncertainty, that commitment to possibility without any guarantee of outcome. The mushroom releases its spores and then dissolves, its work complete, while the spores drift onward, each on its own journey into an unknown future. In every forest, in every ecosystem where plants grow and eventually die, there exists a fundamental problem. The nutrients locked within dead organic matter must be released and made available again for living organisms. Without this recycling, nutrients would accumulate in dead tissue, unavailable to the living, and ecosystems would grind to a halt as available nutrients became depleted. Fungi, particularly the mushroom-forming species, are the primary solution to this problem. The world's most important decomposers, quietly breaking down the dead and returning their components to the cycle of life. When a tree falls in the forest, its wood contains vast quantities of carbon, nitrogen, phosphorus, and countless other elements, all locked within complex organic molecules. The two most abundant components are cellulose and lignin, which together form the bulk of woody tissue. Cellulose is a polymer of glucose molecules, providing structural strength. Lignin is an even more complex polymer that binds cellulose fibers together and makes wood rigid and resistant to decay. Both molecules are difficult to break down, requiring specialized enzymes that most organisms simply do not possess. Fungi have evolved the enzymatic machinery necessary to dismantle these recalcitrant compounds. They secrete powerful enzymes into the wood, enzymes that can break the chemical bonds holding cellulose and lignin together. Different fungi specialize in breaking down different components. Some fungi, called brown rot fungi, primarily decompose cellulose while leaving lignin relatively intact, resulting in wood that becomes brown, crumbly, and cube-like as it decays. Other fungi, called white rot fungi, can break down both cellulose and lignin, bleaching the wood to a lighter color as they remove the lignin. This ability to decompose lignin is particularly remarkable and is found almost exclusively in fungi. Very few bacteria possess this capability, making fungi essential to the carbon cycle in forests. The process of wood decay is slow and methodical. A large fallen tree might take decades to fully decompose, gradually softening, darkening, and breaking apart, eventually crumbling into fragments and then into soil. Throughout this process, fungal mycelium permeates the wood, breaking down cell walls, consuming starches and sugars first, then tackling the more resistant cellulose and lignin. The mushrooms that appear on the log's surface are merely the visible signs of the extensive mycelial network within. As fungi break down dead organic matter, they do more than simply destroy it. They transform it, converting complex organic molecules into simpler forms that other organisms can use. Carbon dioxide is released back into the atmosphere, where plants can use it in photosynthesis. Mineral nutrients are released into the soil, where plant roots and other organisms can absorb them. The fungal mycelium itself becomes food for various soil animals, from microscopic nematodes and springtails to larger invertebrates, further distributing the nutrients through the soil food web. Leaf litter decomposition follows a similar pattern, but on a faster timescale. Leaves that fall in autumn are colonized by fungi almost immediately, their softer tissues more easily broken down than wood. Within months, leaves that were crisp and identifiable become fragmented and indistinct, their substance transformed into humus, that dark, nutrient-rich component of soil that gives it fertility. Fungi are the primary agents of this transformation, working in concert with bacteria and various invertebrates, but doing the heavy lifting of enzymatic breakdown. Different fungal species have preferences for different decay stages and different substrates. Some colonize fresh material, the early decomposers breaking down the most accessible compounds. Others specialize in partially decayed matter, equipped to handle the more resistant components that remain after the early decomposers have taken what they can. This succession of fungal species creates a progression of decay stages, each stage creating conditions for the next, until the original material is completely transformed. The role of fungi in decomposition extends beyond forests to nearly every terrestrial ecosystem. In grasslands, fungi decompose dead grass and herbaceous plants, releasing nutrients for the next year's growth. In agricultural systems, fungi help break down crop residues, though modern farming practices sometimes disrupt these natural processes through tillage and chemical inputs. Even in your garden, fungi are constantly at work, decomposing fallen leaves, dead roots, and other organic matter, maintaining soil fertility without any conscious effort on your part. Some fungi are particularly dramatic in their decomposition roles. The sulfur shelf, or chicken of the woods mushroom, Latiparus sulphureus, grows in large, bright orange and yellow brackets on dead or dying trees, its mycelium rapidly breaking down the heartwood. The artist's conch, Ganoderma applanatum, produces large, woody, shelf-like fruiting bodies that can persist for years, slowly growing as the fungus within continues its patient dismantling of the tree. These perennial fruiting bodies add a new layer of tissue each year, and their undersides can be scratched to create drawings, hence the common name, though doing so damages the fungus. The oyster mushroom, Pleurotus austriatus, grows on dead hardwood trees and has been found to have remarkable decomposition abilities beyond simple wood decay. It can break down various pollutants, including petroleum products and some pesticides, through its enzymatic activity. This has led to research into using fungi for bioremediation, cleaning up contaminated sites by harnessing the fungal ability to break down complex organic molecules. While this technology is still developing, it demonstrates how fundamental fungal decomposition abilities might be applied to human environmental challenges. Decomposition by fungi is not merely destruction, but transformation and renewal. The carbon that was locked in a tree's wood for decades or centuries is released, some as carbon dioxide to the atmosphere, some incorporated into fungal tissue, some left as humus in the soil. That carbon may next become part of a new seedling, or a bacterium, or a passing insect, cycling through the ecosystem in patterns too complex to fully trace. The nitrogen that was in proteins within the tree's cells is broken down and made available again, perhaps taken up by plant roots, perhaps incorporated into microbial biomass, continuing its journey through the living world. Without fungal decomposers, the world would look dramatically different. Dead plant material would accumulate, piling higher year after year, with nutrients locked away in forms living organisms could not access. Forest floors would be choked with undecayed logs and leaves. The carbon cycle would be disrupted. Plant growth would slow and eventually cease as available nutrients became depleted. Fungi prevent this scenario, their patient enzymatic work ensuring that death leads to renewal, that endings provide for new beginnings. The pace of fungal decomposition varies with climate. In warm, moist tropical forests, decomposition is rapid, with fallen leaves and wood breaking down within months to a few years. The forest floor remains relatively clear with little accumulation of debris because fungi and other decomposers work so efficiently in the warm, humid conditions. In cool, dry environments, or in boreal forests where cold temperatures slow biological processes, decomposition is much slower. Fallen logs can persist for decades, and thick layers of partially decomposed organic matter build up on the forest floor. These differences in decomposition rates fundamentally affect how ecosystems function and how nutrients are stored and cycled. Fungi also play essential roles in decomposing animal remains, though this is less often discussed than their role with plant material. When an animal dies, various organisms participate in its decomposition, but fungi are often involved in the later stages, breaking down hair, keratin, and other tough animal proteins. Some fungi specialize in this niche, and their presence can be part of forensic investigations, helping determine time since death based on the succession of fungal species colonizing remains. As we reflect on the role of fungi as decomposers, we might appreciate how death and decay, processes we often find uncomfortable or wish to avoid thinking about, are essential to life's continuation. The mushrooms growing on a rotting log are not signs of corruption but of transformation, not endings but transitions. They remind us that nothing in nature is wasted, that every organism's substance will eventually nourish others, that the boundaries between life and death are more permeable than we sometimes imagine. The fungi stand at that boundary, mediating the passage from one to the other, ensuring that the cycle continues, patient and thorough in their work, asking nothing, simply doing what they have evolved to do, breaking down the complex and making it simple again, closing loops and opening possibilities. In the quiet of the night, as you rest and your thoughts slow, you might imagine the forests around the world, dark and still, where fungi work continuously, invisibly, transforming yesterday's growth into tomorrow's nutrients. where mushrooms release spores that drift through the darkness, where mycelial threads extend molecule by molecule through decaying wood, where the slow work of decomposition proceeds without pause or hurry, a constant renewal happening in the shadows, essential and endless. In the darkness of certain forests on moonless nights when shadows are deepest, a rare and beautiful phenomenon can sometimes be witnessed. Scattered across rotting logs or growing from the forest floor, small mushrooms emit a gentle, ghostly glow, their caps and stems producing a soft greenish light that seems almost supernatural in its quiet radiance. These are bioluminescent mushrooms, fungi that have evolved the ability to produce their own light through chemical reactions within their tissues, creating one of nature's most enchanting displays. Bioluminescence, the production of light by living organisms, is found across many branches of the tree of life. Fireflies flash their signals in summer meadows. Certain fish in the deep ocean carry glowing lures or use light for camouflage. Bacteria can cause seawater to sparkle with blue-green light when disturbed, and various other organisms have independently evolved this capability. Among fungi, approximately 80 species are known to be bioluminescent, though the true number may be higher as many fungi in tropical regions remain poorly studied. The light produced by bioluminescent mushrooms is not bright by human standards. It is a dim glow, barely visible in anything but complete darkness, requiring your eyes to be fully dark adapted to perceive it clearly. The color is typically green to blue-green, with wavelengths in the range of 500 to 530 nanometers, which happens to be near the peak sensitivity of dark-adapted human vision. If you were to encounter these mushrooms in the wild, you would need to find them in complete darkness, away from any artificial light, allowing your eyes time to adjust before the faint luminescence would become visible. The mechanism of fungal bioluminescence involves a chemical reaction between a light-emitting molecule called luciferin and an enzyme called luciferase. When luciferin is oxidized by luciferase in the presence of oxygen, the reaction releases energy in the form of light. This is a cold light, producing virtually no heat, and represents a remarkably efficient conversion of chemical energy directly into photons. The specific luciferin used by fungi was only recently identified by researchers and is different from the luciferins used by fireflies or by bioluminescent marine organisms, indicating that bioluminescence evolved independently in fungi rather than being inherited from a common glowing ancestor. Not all parts of a bioluminescent mushroom necessarily glow with equal intensity. In some species, the mycelium produces light, but the mushroom fruiting body does not. In others, the mushroom glows, but the mycelium in the wood or soil does not. Some species have glowing caps and stems, while in others only certain structures produce light. This variation suggests that the light serves different functions in different species, or that the trait is being gained or lost over evolutionary time in ways that are not yet fully understood. One of the most well-known bioluminescent species is Pinellus stypticus, a small mushroom that grows in overlapping clusters on decaying hardwood logs across much of North America, Europe, and Asia. Not all populations of this species are bioluminescent, however. In North America, particularly in eastern regions, Pinellus stypticus produces visible light, but European populations of the same species typically do not glow, or glow only very faintly. This geographic variation in a single species adds another layer of mystery to the phenomenon. Another striking bioluminescent species is Omphalotus olearius, commonly called the jack-o'-lantern mushroom due to its orange color and its glow. This species grows on dead hardwood, often appearing to grow from the ground but actually connected to buried roots or wood. It is poisonous if eaten, causing severe gastrointestinal distress, but it is not deadly. Its bioluminescence is among the brightest of fungal bioluminescence, though still quite dim by objective standards. The gills on the underside of the cap produce most of the light, giving the mushroom an eerie appearance in complete darkness. In tropical regions, several species in the genus Mycelium produce bioluminescence. These are small, delicate mushrooms with thin stems and caps, growing on leaf litter or dead wood in humid forests. Their glow is faint, visible only in the deepest darkness of the forest at night. Some tropical bioluminescent species grow directly on living trees, attached to bark, their small glowing caps creating tiny points of light scattered up tree trunks. The evolutionary purpose of bioluminescence in fungi has been debated. In fireflies, the light clearly serves as a mating signal. In deep-sea fish, it can serve for communication, camouflage, or attracting prey. But what benefit do fungi gain from producing light? Fungi do not have eyes and cannot see their own light. They do not use it for hunting or escaping predators. So why expend energy on this seemingly purposeless glow? Recent research suggests that the light may attract nocturnal insects, which then help disperse the fungal spores. Studies comparing bioluminescent and non-bioluminescent mushrooms found that when artificial light of the same wavelength as fungal bioluminescence was shown on non-glowing mushrooms at night, more insects visited those illuminated mushrooms compared to dark controls. This suggests insects are attracted to the light, and once on the mushroom, they may pick up spores on their bodies and carry them to new locations. This would make fungal bioluminescence analogous to flowers that attract pollinators, except fungi are attracting spore dispersers through light rather than fragrance or nectar. However, this hypothesis does not fully explain bioluminescence in all fungal species, particularly those where the mycelium glows but the spore-producing fruiting body does not. Alternative explanations include that bioluminescence might be a metabolic byproduct, a side effect of other biochemical processes that happens to produce light without necessarily serving an adaptive function, or that it once served a function that is no longer relevant but the trait persists. The truth may vary among species, with light serving different functions or no function depending on the particular ecology and evolutionary history of each bioluminescent lineage. The experience of witnessing bioluminescent mushrooms in their natural habitat is, by all accounts, memorable and somewhat otherworldly. Imagine walking through a tropical forest at night, the darkness so complete you can barely see your own hand, and gradually becoming aware of tiny points of soft green light scattered across the forest floor or glowing on the side of a tree trunk. The light does not flicker like fireflies, but glows steadily, a constant gentle luminescence that seems to come from within the fungal tissue itself. There is something both eerie and beautiful about it. Life producing light in the darkness, a quiet assertion of presence in the depths of night. Bioluminescent mushrooms have inspired folklore and stories in cultures around the world. Mysterious glows in forests at night, patches of luminous decay on old stumps. These would have been unsettling phenomena to people without scientific understanding of their cause. Various supernatural explanations arose. Stories of fairies or spirits, of ghostly presences in the woods. Even the term foxfire, used to describe the glow of luminous wood colonized by fungal mycelium, hints at folkloric associations suggesting something magical or tricksterish about the phenomenon. Today, we understand that bioluminescent mushrooms operate through chemistry rather than magic. But understanding the mechanism does not diminish the wonder. If anything, knowing that life has found ways to convert chemical energy directly into light, that fungi growing on a rotting log in complete darkness can glow with their own generated luminescence, deepens the sense of nature's creativity and possibility. These mushrooms remind us that the world contains phenomena outside our everyday experience, that nature includes subtleties and rarities that require specific conditions and patient observation to witness. The dim light of bioluminescent mushrooms also reminds us of scale and sensitivity. In a world increasingly dominated by artificial light where true darkness is rare and night skies wash out in light pollution, the ability to perceive such faint natural light becomes increasingly uncommon. To see these mushrooms glow requires not just finding them but also accepting complete darkness, allowing your eyes to adapt, becoming comfortable in an environment where sight is limited. It requires a kind of patience and stillness that runs counter to modern life's pace and intensity. In this way, the mushrooms offer not just a visual experience, but an invitation to a different way of being, quieter and more receptive, attuned to subtleties rather than constantly stimulated by intensity. As your mind considers these glowing fungi and dark forests, imagine that soft greenish light, steady and gentle, asking nothing, simply glowing because its chemistry allows it, a small assertion of the living world's capacity for beauty and strangeness. Let that image settle into your thoughts as you drift towards sleep, a reminder that even in darkness there are sources of light, quiet and persistent, neither bright nor demanding, but present nonetheless, glowing through the night. Beyond the general biology and ecology of mushrooms lie specific phenomena that capture the imagination through their strangeness, their beauty, or their scientific intrigue. These are the curiosities and specializations, the adaptations that seem almost designed to make us pause and reconsider what we thought we knew about fungi. Tonight we will explore a few of these phenomena, each revealing something about the remarkable diversity and ingenuity of fungal life. Fairy rings are among the most visually striking patterns that mushrooms create. These are rings of mushrooms that appear in lawns or meadows, growing in nearly perfect circles that can range from just a few feet to dozens of yards in diameter. The name comes from European folklore, which attributed these rings to dancing fairies or elves, their circular dances marking the ground and causing mushrooms to grow where they stepped. Various superstitions surrounded fairy rings, some considering them lucky, others seeing them as dangerous places where humans might be enchanted or led astray. The actual explanation is less magical but equally fascinating from a biological perspective. A fairy ring begins when a single spore germinates and the resulting mycelium begins to grow outward in all directions from that central point. As the mycelium expands radially, it depletes nutrients in the soil near the center while continuing to grow at the outer edge, forming an ever-expanding circle. The mushrooms appear at this outer growing edge, where the mycelium is most active and conditions trigger fruiting body formation. Year after year, the ring expands outward, the mycelium at the center dying back or becoming inactive while the periphery continues its growth. The rate of expansion varies by species and environmental conditions, but many fairy rings grow outward at a rate of a few inches to a foot per year. By measuring the diameter of a ring and knowing the typical expansion rate for that species, researchers can estimate the age of the mycelial colony. Some fairy rings are believed to be centuries old, the same individual fungus growing and expanding since before modern history, creating larger and larger circles as time passes. There is something quietly impressive about this patient, persistent growth, a living organism marking its existence through geometry, creating patterns visible from above, expanding imperceptibly but unstoppably through time. In lawns, fairy rings sometimes cause problems for homeowners. The mushrooms themselves are often considered unsightly, and the mycelial activity in the soil can affect grass growth. Sometimes the grass at the ring's edge grows more vigorously and greener than surrounding grass, stimulated by nutrients released by fungal activity. Other times the grass dies in a ring pattern, suffocated by dense mycelial mats that make the soil hydrophobic, unable to absorb water. These variations depend on the fungal species and soil conditions. But from a purely biological perspective, the fairy ring represents an elegant strategy for resource exploitation. The fungus systematically harvesting nutrients from ever-expanding territory. Truffles represent another remarkable fungal adaptation, a complete abandonment of above-ground dispersal in favor of an underground existence, while most mushrooms elevate their spores into air currents for dispersal. Truffles remain entirely subterranean, producing their spore-bearing structures beneath the soil surface. These structures are often irregular, lumpy, and completely enclosed, protecting the spores within. The challenge for a truffle is obvious. If your spores are buried underground, how do you disperse them? The solution is to attract animals that will dig you up, eat you, and deposit your spores elsewhere in their feces. To accomplish this, truffles have evolved to produce some of the most powerful and distinctive aromas in the fungal kingdom. These aromas are complex mixtures of volatile organic compounds, and the human nose finds many of them intensely appealing, which is why truffles are so prized in cuisine. The specific compounds responsible for truffle aroma vary by species, but many include sulfur-containing molecules that produce earthy, musky, garlicky notes, as well as alcohols and other volatiles that add additional layers of scent. Some of these compounds are similar to mammalian pheromones, which may explain why animals, particularly pigs, are so strongly attracted to truffles. Dogs can also be trained to find truffles by scent, and truffle hunters historically have used both pigs and dogs to locate these buried treasures. From the truffle's perspective, a strong aroma that attracts animals is essential for reproduction. Animals dig up the truffle, consume it, and the spores pass through the digestive system intact. Hours or days later, when the animal defecates elsewhere, the spores are deposited in a new location along with a package of fertilizer, giving them an excellent start. This strategy has proven successful enough that numerous fungal species have independently evolved the truffle growth form and underground lifestyle. The underground lifestyle also protects truffles from desiccation and temperature fluctuations, allowing them to fruit during seasons when above-ground conditions might be unfavorable. Some truffle species fruit in winter when few above-ground mushrooms are active, taking advantage of the soil's temperature buffering. The evolutionary trade-off is that truffles must invest heavily in aroma production to ensure dispersal, whereas mushrooms that grow above ground can rely on passive dispersal by wind and need not produce strong scents. Moving from the hidden world of truffles to the ancient past, fossil mushrooms provide glimpses into fungal evolution. Fungi rarely fossilize because their fruiting bodies are soft and decay rapidly, and even mycelium in soil or wood typically leaves no trace in the fossil record. However, under exceptional preservation conditions, fungal structures can be preserved, offering precious windows into the deep history of these organisms. The oldest confirmed mushroom fossil was discovered in a piece of amber from Myanmar, dated to approximately 100 million years ago, from the Cretaceous period when dinosaurs still walked the earth. This fossil preserves a small mushroom cap attached to a piece of wood, the details so fine that gill structures and other features remain visible. The mushroom closely resembles modern members of the Agaricales order, suggesting that many mushroom forms had already evolved by this time. Even more remarkably, the same piece of amber contained other fossils, including a beetle that appeared to have been feeding on the mushroom. This provides direct evidence that the ecological relationships we see today with insects interacting with mushrooms were already established 100 million years ago. The mushroom, the beetle, the tree from which the resin flowed, all captured together in amber, frozen in a moment that occurred in the age of dinosaurs. Older fungal fossils exist, but these are typically preserved as microscopic structures, spores or high-fall fragments, or as evidence of fungal interactions with plants rather than as recognizable mushrooms. Molecular clock studies, which use genetic mutation rates to estimate when lineages diverged, suggest that fungi as a kingdom are much older than the mushroom fossil record indicates, potentially dating back over 1 billion years. Mushroom-forming fungi likely arose hundreds of millions of years ago, but direct fossil evidence remains scarce due to preservation biases. Some fungi have evolved striking morphologies that seem almost designed for drama. The stinkhorn fungi, members of the Phyllaceae family, produce fruiting bodies that emerge rapidly from egg-like structures, extending upward in shapes that range from fingers to lattices to bizarre cage-like forms. Many have a slimy, foul-smelling spore mass at the tip or covering parts of the structure. The smell, often compared to rotting meat or feces, attracts flies, which feed on the slime and in doing so coat themselves with spores. As the flies move to other locations, they disperse the fungal spores. The strategy is effective but aesthetically challenging for human sensibilities, a reminder that fungi evolved to succeed in their own ecological contexts, not to please human observers. The bird's nest fungi represent another unusual morphology. These small fungi produce cup-shaped fruiting bodies that resemble tiny nests containing eggs. The eggs are actually spore packets called peridioles. When raindrops hit the cup with sufficient force, the peridioles are splashed out, sometimes traveling several feet from the parent structure. Some species have peridioles attached to sticky cords that can wrap around nearby vegetation, ensuring the spore packet remains elevated above ground where it can be further dispersed by passing animals or additional rain. This splash cup dispersal mechanism is elegant in its simplicity, using the energy of falling raindrops to launch spores without requiring any active mechanism by the fungus itself. The variety of forms and strategies among fungi reflects millions of years of evolution, each lineage adapting to its particular ecological niche, solving the challenges of nutrition acquisition, growth, and reproduction in different ways. Some solutions are widespread, evolved repeatedly like the mushroom form itself, which provides an elevated platform for spore dispersal. Other solutions are rare and specialized, found in just a few species occupying narrow ecological niches. Together, this diversity creates a kingdom of life that is endlessly fascinating in its variations on fungal themes. As we contemplate these phenomena, fairy rings expanding slowly through the years, truffles luring animals with their complex perfumes, ancient mushrooms preserved in amber, stink horns attracting flies with their putrid scent. We can appreciate how evolution produces not elegance in any universal aesthetic sense, but rather fitness, solutions that work in context, strategies that succeed in allowing organisms to survive and reproduce. The mushroom kingdom is full of examples that strike us as beautiful, strange, disgusting, or ingenious. But to the fungi themselves, these are simply ways of being, forms that have persisted because they function, because they solve the fundamental problems of existence in their particular environments. Let these images rest in your mind as your breathing slows and deepens. Circles of mushrooms expanding through grass, truffles hidden in soil, their presence announced only by scent, ancient forests where mushrooms grew beneath dinosaur feet, a world of forms and strategies, quiet and persistent, operating according to biological principles that are both comprehensible and endlessly creative in their specific implementations. There is a kind of peace in understanding that life finds ways, that existence manifests in countless forms, that beneath the surface of familiarity lies depth and strangeness and adaptation beyond what we commonly notice. Mushrooms are chemical factories producing a remarkable array of compounds that serve various functions in the life of the fungus. Some of these chemicals defend against predators or competitors. Others facilitate communication or regulate development. Many have properties that affect other organisms, including humans, and some have found their way into traditional medicine and modern pharmacology. Tonight we will explore the chemistry of mushrooms, the molecular basis of their effects and interactions, approaching these topics from a scientific perspective that emphasizes understanding over sensation. The fundamental structure of fungal cells differs from plant and animal cells in important ways. While plant cell walls are primarily made of cellulose, fungal cell walls are constructed mainly from chitin, the same polysaccharide that forms the exoskeletons of insects and crustaceans. Chitin is a polymer of N-acetylglucosamine, arranged in long chains that are cross-linked to form strong, flexible sheets. This chitin-based architecture gives fungal cells their structural integrity and protection. The presence of chitin in fungal cell walls has several implications. First, it makes fungi more closely related to animals than to plants in evolutionary terms, a fact that was not fully appreciated until molecular phylogenetic studies confirmed it. Second, chitin is difficult for most organisms to digest, providing fungi with protection from potential predators. Third, when humans consume mushrooms, the chitin in cell walls can make some nutrients less accessible, which is why cooking mushrooms, which breaks down cell walls and cell membranes, generally makes them more digestible and their nutrients more bioavailable. Beyond structural components, mushrooms produce thousands of secondary metabolites, compounds that are not essential for basic growth and reproduction but serve other functions. Many of these are defensive compounds, deterring animals from eating the mushrooms or preventing bacterial and fungal competitors from invading the mycelium. Some mushrooms are highly toxic to humans and other mammals, producing compounds that can cause liver failure, kidney damage, neurological effects, or gastrointestinal distress. The death-cap mushroom Amanita phalloides produces amatoxins, cyclic peptides that inhibit RNA polymerase II, an enzyme essential for transcription in eukaryotic cells. When consumed, even a single mushroom can be fatal, as the toxins cause progressive liver and kidney failure over several days. There is no antidote, and treatment is largely supportive, hoping the body can survive while clearing the toxins. This mushroom accounts for the majority of fatal mushroom poisonings worldwide, partially because it can resemble edible species and partially because its toxins do not cause immediate symptoms, delaying recognition of poisoning until significant damage has occurred. Other mushrooms produce different classes of toxic compounds. Some contain muscarine, which affects the parasympathetic nervous system, causing excessive salivation, sweating, and potentially dangerous effects on heart rate and breathing. Others produce compounds that cause gastrointestinal upset without being life-threatening. Still others contain toxins that destroy red blood cells or cause neurological symptoms. The diversity of fungal toxins reflects the various ecological pressures fungi face and the different chemical solutions evolution has found for deterring predation. Not all biologically active compounds in mushrooms are harmful. Many species produce compounds with medicinal properties that have been used in traditional medicine systems for centuries and are now being studied scientifically. The medicinal mushrooms include species like reishi, Organoderma lucidum, which produces triterpenes and polysaccharides that appear to have anti-inflammatory and immune-modulating effects. Shiitake Lentinula edodes contains lentinan, a beta-glucan that has been investigated for its potential to enhance immune function. Turkey tail Trametes versicolor produces polysaccharide K, which has been approved as an adjunct cancer treatment in Japan and is undergoing studies elsewhere. The mechanisms by which these compounds act are still being elucidated, but many appear to interact with immune system components, particularly affecting the activity of various white blood cells. The polysaccharides in many medicinal mushrooms are thought to act as immunomodulators, helping to regulate immune responses rather than simply stimulating or suppressing them. This is an area of active research, with many compounds showing promise in laboratory and animal studies, though rigorous human clinical trials are still needed for most applications. One class of fungal compounds that has received significant scientific attention in recent years is the psilocybin and related tryptamine alkaloids found in certain mushroom species, particularly those in the genus Psilocybe. Psilocybin is converted in the body to psilocin, which acts as an agonist at serotonin receptors, particularly the 5-HT2A receptor in the brain. This interaction produces profound changes in consciousness, perception, and mood, effects that have been known to indigenous cultures for millennia and have been investigated scientifically since the mid-20th century. Recent research using brain imaging techniques has shown that psilocybin disrupts the normal patterns of communication between brain regions, temporarily reducing the strong connections within established networks and allowing increased communication between regions that do not typically interact. This disruption of normal brain network organization correlates with the subjective effects reported by individuals, including altered sense of self, changes in sensory perception, and shifts in emotional processing. The effects are temporary, with brain activity returning to baseline patterns within hours. But studies suggest that even single experiences can produce lasting changes in personality measures, particularly increases in openness, and can have therapeutic benefits for conditions like treatment-resistant depression and anxiety associated with serious illness. The mechanism appears to involve neuroplasticity, the brain's ability to form new connections and patterns. By temporarily disrupting established patterns, psilocybin may allow the formation of new neural pathways and can help individuals break out of rigid patterns of thought and emotion that characterize certain psychiatric conditions. This is an active area of research with multiple clinical trials underway examining therapeutic applications, though it is important to note that these investigations are taking place in controlled clinical settings with careful screening, preparation, and support, quite different from recreational use. From a purely biochemical perspective, the fact that a molecule produced by a mushroom can so profoundly affect human consciousness raises interesting questions about the evolution of these compounds and their natural functions. Psilocybin-containing mushrooms are typically found growing on dung or in disturbed soils rich in organic matter. The compounds may serve defensive functions, deterring insects or other invertebrates from consuming the mushrooms, or they may be metabolic byproducts without specific adaptive function. The human effects are likely incidental from the mushroom's perspective, but they have given these species profound cultural significance in various human societies. Mushrooms also produce numerous enzymes with interesting properties and potential applications. The ligninolytic enzymes that white rot fungi use to break down lignin in wood are being investigated for industrial applications, potentially useful in processing plant biomass for biofuels or in paper production. Lacases and peroxidases from fungi can oxidize various compounds and are being explored for bioremediation applications and even in biosensors. The chitinases that some fungi produce to break down the chitin in competitors' cell walls have potential applications in agriculture and medicine. The chemistry of mushrooms extends to their nutritional composition as well. Most mushrooms are low in calories, containing mostly water, but they provide protein, B vitamins, particularly niacin and riboflavin, minerals like selenium, copper, and potassium, and various antioxidants. Some species, particularly those exposed to ultraviolet light, can synthesize vitamin D2, making them one of the few non-animal food sources of this vitamin. The specific nutritional profile varies by species and growing conditions. The flavors and aromas of edible mushrooms also arise from their chemistry. The umami taste, that savory, meaty flavor quality, comes primarily from glutamate and related compounds that mushrooms produce in significant quantities. Various volatile compounds contribute to mushroom aroma, including 8 carbon compounds that give mushrooms their characteristic earthy smell, and sulfur-containing compounds in some species that can produce garlic-like or other distinctive notes. The chemistry of flavor is complex, with dozens or hundreds of compounds contributing to the overall sensory experience of consuming any particular mushroom species. As we consider the chemistry of mushrooms, we can appreciate that these organisms are not passive structures, but are actively producing compounds that mediate their interactions with the world. Every molecule serves some function in the fungal life cycle, even if that function is not immediately obvious to us. The toxins protect, the enzymes digest, the signaling molecules communicate, the structural compounds provide support and protection, and occasionally, through the contingencies of evolution and biochemistry, some of these fungal compounds interact with human biology in ways that we find useful, harmful, therapeutic, or consciousness-altering. This molecular complexity reminds us that even simple-seeming organisms are elaborate at the chemical level, that the mushroom you might pass on a walk contains within its cells a pharmaceutical factory producing dozens or hundreds of compounds, each with its own structure and properties, each the result of genes shaped by millions of years of evolution. There is something humbling in that complexity, a reminder that understanding even a single mushroom completely would require knowledge spanning genetics, biochemistry, ecology, and evolution, that simple appearances can mask profound depths. Let your thoughts settle around these chemical concepts, these molecules and mechanisms, understanding that beneath the visible forms of mushrooms lies this invisible molecular realm where the actual work of life proceeds. where enzymes catalyze reactions, where toxins defend, where signals communicate, all happening continuously, molecule by molecule in the quiet darkness within every fungal cell. The fungi we recognize as mushrooms represent just a fraction of the kingdom fungi, but even within this subset, diversity is staggering. Scientists estimate that there may be millions of fungal species on Earth, though only a small percentage have been formally described and named. The mushroom-forming species, those that produce visible fruiting bodies, number in the tens of thousands, displaying remarkable variation in form, ecology, and life strategy. Tonight we will explore how scientists organize this diversity, the major groups of mushroom-forming fungi, and the principles that guide fungal classification. Classification systems attempt to organize life's diversity in ways that reflect evolutionary relationships. Modern fungal classification relies heavily on DNA sequencing and molecular phylogenetics, comparing genetic sequences among species to determine which are most closely related, and constructing evolutionary trees that show how lineages diverged from common ancestors. This molecular approach has revolutionized fungal taxonomy, revealing relationships that morphology alone could not clarify and sometimes overturning classifications that had stood for decades. The kingdom fungi encompasses multiple major divisions or phyla. The two most important for our consideration are Basidiomycota and Ascomycota, as these include the vast majority of mushroom-forming species. However, there are other fungal phyla, including Zygomycota, which contains many common molds, and Chytridiomycota, primitive fungi that produce motile spores with flagella, and are mostly aquatic or soil-dwelling. These groups rarely produce anything we would recognize as a mushroom, though they are diverse and ecologically important in their own right. Basidiomycota, the Basidiomycetes or club fungi, produce their spores on club-shaped cells called basidia. Each basidium typically produces 4 spores on small projections called sterigmata. The classic mushrooms with gills or pores, the shelf fungi that grow from trees, the puffballs, earth stars, stinkhorns, and jelly fungi all belong to Basidiomycota. This phylum contains approximately 30,000 described species, but the true number is certainly much higher. Basidiomycetes include some of the most economically important fungi, from edible mushrooms like the common button mushroom Agaricus bisporus, to plant pathogens like the rusts and smuts that affect agricultural crops. Within Basidiomycota, the order Agaricales contains the typical gilled mushrooms, those with a cap, stem, and radiating gills on the underside of the cap. This enormous order includes familiar edible species like shiitake and oyster mushrooms, deadly poisonous species like the death cap and destroying angels, hallucinogenic psilocybin mushrooms, and countless species that are neither particularly useful nor harmful to humans but play important roles in forest ecology. The sheer diversity within Agaricales is testimony to how successful this basic body plan has been evolutionarily. Another major order within Basidiomycota is Boletales, which includes the boletes, mushrooms that have pores rather than gills on the cap undersurface. These pores are the openings of vertical tubes in which spores are produced. Boletes are mostly mycorrhizal, forming partnerships with tree roots, and many are choice edibles, including the prized porcini or king bolete, Boletus edulis. The spongy pore surface distinguishes boletes from gilled mushrooms at a glance, though the evolutionary relationship between these groups is complex, with some gilled species actually being more closely related to certain boletes than to other gilled mushrooms. The Polyporales, another basidiomycete order, includes most of the shelf or bracket fungi that grow on wood, producing tough, leathery or woody fruiting bodies that can persist for years. These fungi are crucial decomposers of woody material, breaking down fallen logs and standing dead trees. Some species in this group are cultivated for medicinal purposes, including reishi and turkey tail. The durability of polypore fruiting bodies contrasts sharply with the ephemeral nature of most gilled mushrooms, which appear and decay within days. Ascomycota, the ascomycetes or sac fungi, produce their spores inside sac-like structures called asci. Each ascus typically contains 8 spores arranged in a row. This phylum is incredibly diverse, containing approximately 65,000 described species, including not just mushrooms but also yeasts, molds, and various microscopic fungi. The mushroom-forming ascomycetes include morels, false morels, cup fungi, and truffles, species whose fruiting bodies are often quite different in structure from basidiomycete mushrooms. Morels, genus Morchella, are among the most prized edible fungi, with their distinctive honeycombed caps composed of pits and ridges. These mushrooms appear in spring, often in association with disturbed ground, burned areas, or dying trees. The specific ecology of many morel species remains poorly understood, adding to their mystique and making cultivation challenging, though some species have been successfully brought into commercial production in recent years. Truffles, which we discussed earlier, are ascomycetes that have completely abandoned above-ground dispersal. Multiple truffle lineages have evolved independently within Ascomycota, an example of convergent evolution where similar ecological pressures lead to similar solutions in different evolutionary lineages. The most valuable culinary truffles, including white truffles, tuber magnatum, and black Perigord truffles, tuber melanosporum, are ascomycetes growing in mycorrhizal association with tree roots. The diversity of forms within fungi includes many that challenge our intuitions about what a mushroom is. Jelly fungi produce gelatinous fruiting bodies that can dry out and rehydrate repeatedly. Coral fungi form branching structures that resemble undersea corals. Bird's nest fungi, as mentioned earlier, create tiny cup-shaped structures containing spore packets. Earth tongues produce club-like projections from the ground. The variety reflects the many different solutions evolution has found to the basic challenge of producing and dispersing spores. Classification below the level of order becomes increasingly detailed, with families, genera, and species reflecting finer distinctions in characteristics and relationships. Species identification in fungi can be challenging, requiring careful attention to multiple features: the size, shape, and color of the fruiting body, the structure of gills or pores, the presence or absence of veils, rings, or volvas, the color and texture of the spore print made by allowing spores to fall from a cap onto paper, microscopic examination of spore shape and size, and increasingly, DNA sequence analysis. Many mushrooms that appear similar are actually distinct species, while others show considerable variation in appearance despite being the same species. The estimate of millions of potential fungal species comes from various sampling studies and extrapolation methods. When researchers intensively sample fungi in particular habitats using both traditional collection and environmental DNA techniques, they consistently find many more species than were previously known from those areas. By comparing the ratio of known to newly discovered species and applying this to global estimates of habitat diversity, mycologists arrive at the conclusion that the vast majority of fungal diversity remains undescribed. Each patch of forest, each meadow, each ecosystem likely contains fungal species unknown to science, living quietly in soil or wood, perhaps rarely producing visible fruiting bodies, their existence unrecognized. This hidden diversity has practical implications. Among those unknown species may be fungi with novel enzymes useful in industry, with compounds that could become medicines, with ecological roles that we have not yet understood. As habitats are lost to development and climate change, species go extinct before they are ever known. The urgency of cataloging fungal diversity grows as the environments that harbor that diversity face increasing pressures. Citizen science initiatives have become increasingly important in documenting fungal diversity. Websites and apps allow amateur naturalists to photograph mushrooms and submit observations which experts can then help identify. These crowdsourced data sets are revealing patterns in fungal distribution, phenology, and abundance that would be impossible to document through traditional scientific surveys alone. The observations of thousands of people walking through forests and fields, noticing mushrooms and recording what they find, collectively build a picture of fungal diversity across landscapes and seasons. Understanding fungal classification helps us appreciate that when we see a mushroom, we are seeing the representative of a lineage, a branch on the evolutionary tree of fungi that diverged from other branches millions of years ago and has been evolving independently ever since. That mushroom's shape and color, its ecology and chemistry all reflect its evolutionary history and the particular solutions its lineage developed to the challenges of survival and reproduction. Each species is a unique combination of traits, a point in the vast space of possible fungal forms. As your mind contemplates this diversity, these thousands of species and millions more unknown, imagine the forests and fields of the world, each with its own fungal community, species sorted by climate and habitat, by soil type and available partners, forming communities where decomposers and mycorrhizal species and pathogens all coexist, competing and cooperating, each playing its role in the larger ecological system. Imagine the work of taxonomists, carefully describing species, comparing specimens, sequencing DNA, slowly building the catalog of fungal life, adding species to the known inventory one by one, aware that the task is immense and may never be truly complete. There is something both humbling and comforting in that vastness, in knowing that so much remains unknown, that the world contains multitudes of organisms going about their lives unnoticed by human science. That complexity and diversity extend beyond our current knowledge and perhaps beyond what we will ever fully document. The mushrooms we see are ambassadors from that larger realm, visible representatives of a kingdom mostly hidden, inviting us to appreciate depth and diversity, to remain curious and humble in the face of nature's scope. We have traveled together through the world of mushrooms, exploring their biology, ecology, chemistry, and diversity. From the vast underground networks of mycelium to the microscopic journey of spores, from glowing mushrooms in dark forests to fairy rings expanding through the centuries, we have encountered a kingdom of life that operates according to principles both familiar and strange, organisms that are neither plant nor animal, but something distinctly their own. Mushrooms remind us that the visible world is just a fraction of what exists. The fruiting bodies we see are temporary expressions of organisms that persist mostly out of sight. Networks threading through soil and wood, partnerships connecting trees across forests, chemical factories producing compounds that shape ecosystems and affect other organisms in countless ways. To understand mushrooms requires looking beneath surfaces, considering what is hidden, appreciating that the most important aspects of their lives occur in darkness and silence, slowly and continuously. The interconnectedness that mycorrhizal networks represent extends beyond forests. Fungi connect the living to the dead through decomposition, recycling nutrients from what has ended back into what continues. They connect plants to soil, extracting minerals that roots alone could not access. They connect present to past through their evolutionary history, carrying forward solutions developed millions of years ago. And they connect us as observers and students to the fundamental processes that make ecosystems function, that make life on land possible. There is wisdom in the fungal way of being if we choose to see it. Patience as mycelium expands molecule by molecule, year by year, without haste or anxiety. Adaptability as fungi colonize nearly every environment on Earth, from Arctic tundra to tropical rainforests, from desert soils to the depths of caves. Cooperation as mycorrhizal partnerships demonstrate mutual benefit arising from exchange rather than exploitation. Persistence as fairy rings expand through centuries and the humongous fungus grows through millennia. Transformation as decomposition turns death into the raw materials for new life. Mushrooms also remind us of the limits of our knowledge and the value of humility. Millions of species remain undescribed. The ecological roles of even known species are often incompletely understood. The chemistry of fungal compounds continues to surprise researchers. The complexity of soil fungal communities and their interactions with plants and other organisms challenges our ability to model and predict ecosystem behavior. We have learned much, but vast territories of ignorance remain, inviting continued curiosity and study. As you rest now, allowing your thoughts to settle and your body to relax completely, you might carry with you a few images from our journey. Picture mycelium spreading through dark soil, threads too fine to see individually but collectively covering acres. Picture spores drifting on invisible air currents, each one a possibility, a potential beginning. Picture trees connected underground, sharing resources through fungal intermediaries. The forest functioning as a community rather than a collection of isolated individuals. Picture a mushroom glowing faintly in complete darkness, its soft green light neither bright nor demanding, simply present. These images need not carry any particular meaning or message. They are simply aspects of the natural world, phenomena that occur whether we observe them or not, processes that have continued for millions of years before humans existed and will continue long after we are gone. There is something peaceful in that perspective, in recognizing that we are part of something much larger and older, that the world contains wonders and complexities operating at scales from microscopic to landscape spanning, from moments to millennia. The next time you see a mushroom, perhaps you will pause and consider it more deeply. Not just the visible cap and stem, but the mycelium beneath, the spores it is releasing, the nutrients it is cycling, the partnerships it may be maintaining, the evolutionary history it carries, the chemistry occurring within its cells. You might appreciate it not just as an object, but as a process, a moment in a life cycle. An organism actively engaged in its existence, feeding and growing and reproducing according to strategies refined over countless generations. And if you walk in a forest, you might think about what lies beneath your feet. The mycorrhizal networks connecting tree to tree, the decomposers breaking down fallen logs and leaf litter. The quiet work of fungi proceeding invisibly, supporting the visible forest, making possible the trees and flowers and all the life that depends on them. The forest above and the fungal realm below, each shaping the other, interdependent and interconnected in ways both profound and subtle. This has been a journey through sleepy science facts about mushrooms, an exploration conducted slowly and calmly, with space for ideas to settle and understanding to develop gently. We have covered the major aspects of fungal biology and ecology, the phenomena that make mushrooms fascinating subjects of study, the diversity that makes each species unique, and the connections that link fungi to broader patterns in nature. The information is here for you to remember or to let drift away as you prefer. No pressure to retain every detail, simply an offering of knowledge for your consideration. If you found yourself learning something new tonight, something that shifted your understanding or sparked your curiosity, then this exploration has served its purpose. If you found your mind wandering or drifting towards sleep while these facts and stories unfolded, that too is perfectly acceptable. The intention has been to provide a calm, informative journey suitable for rest and relaxation, knowledge presented without urgency or intensity, facts shared in the spirit of gentle education rather than demanding memorization. Before we close, a quiet suggestion. If you enjoyed this exploration of the fungal kingdom and found value in this slow, thorough approach to science education, consider subscribing to this channel for more journeys through the natural world and scientific concepts, presented in this same calm, sleep-friendly style. And if you found this particular video helpful or interesting, a like would be appreciated, as it helps others discover this content. These simple actions support the creation of more material like this, allowing these explorations to continue. But these requests are gentle, without pressure. Whether you subscribe or not, whether you like this video or simply close it and move on, the knowledge shared tonight remains. The facts about mushrooms and fungi exist independent of any particular response, and the natural world continues its processes regardless of our engagement or attention. There is no obligation here, only invitation, only possibility. As we reach the end of our time together tonight, let yourself settle fully into rest. Let the images and information from this journey drift through your mind without trying to hold on to them. Let your breathing deepen and slow. Let your body release any remaining tension, sinking into whatever surface supports you, becoming heavy and relaxed and still. The world of mushrooms will continue without your attention. Mycelium growing, spores dispersing, decomposition proceeding, networks connecting. All the quiet work of fungi happening in darkness and silence, patient and persistent and unhurried. Thank you for joining this exploration. May your rest be deep and your dreams, if any, be peaceful. Good night.